Hello students, today we review on lipid metabolism. Mainly we will discuss about fatty acids, beta oxidation. So cells always prefer to utilize glucose and amino acids to produce energy when there is no option and the cell is not in hypoxic condition. Cellular glucose and ATP level is very low. Cells of cardiac muscles, skeletal muscles, smooth muscles, liver, renal cortex, brown adipose tissue, etc. start to oxidize fatty acids to produce acetyl-CoA. This acetyl-CoA undergo tricarboxylic acid cycle to produce NADH and FADH2, which by electron transport chain produces ATP. Acetyl coenzyme A in liver also can produce ketone body for gluconeogenesis to supply glucose in brain and RBC during that time. So, free fatty acids bind with albumin in blood and transported to the liver, muscles, kidney, and many other tissues for metabolism to produce energy, except brain, because it could not cross the blood-brain barrier, and RBC also could not utilize fatty acids because it has no mitochondria. Fatty acids have to be activated to acyl coenzyme A and then transported to mitochondria prior to oxidation. In presence of ATP and coenzyme A, acyl-CoA synthetase catalyzes the esterification of free fatty acids to fatty acyl-CoA. This reaction is called activation of fatty acids. In this reaction, a thioester linkage is formed between the fatty acid carbon group and the thiol group of coenzyme A. The ATP undergoes pyrophosphate cleavage to yield AMP and inorganic pyrophosphate. Acyl-CoA synthetase is present in outer membrane of mitochondria, endoplasmic reticulum, and peroxisome. There are different types of acyl-CoA synthetase specific for their different fatty acids for different chain length. Acetyl-CoA synthetase activates acetic, propionic, and acrylic acids. Medium chain acyl-CoA synthetase activates 4 to 12 carbon fatty acids. Long chain acyl-CoA synthetase activates 12 to 24 carbon fatty acids. They can activate saturated, unsaturated, and hydroxy fatty acids also. After activation, short-chain fatty acids can diffuse freely inside the mitochondrial matrix, but long-chain fatty acids, that is about 14 to 20 carbon fatty acids, enter into the mitochondria by the acyl carnitine shuttle. Very long-chain fatty acids, that is 22, 20, 20 or 24 carbon fatty acids, are oxidized in peroxisomes. Long chain fatty acids, that is 14 to 20 carbon fatty acids, can cross the outer mitochondrial membrane through porine channels but could not cross the inner mitochondrial membrane. Scientists I.B. Fridge and others showed that the stimulation of fatty acid oxidation by carnitine is due to the action of an enzyme, carnitine acyltransferase 1, present in the outer mitochondrial membrane. Carnitine or beta-hydroxy gamma trimethyl ammonium butyrate forms oxygen ester linkage between its hydroxyl group and fatty acyl group of fatty acyl coenzyme A. Carnitine acylcarnitine translocase present in the inner mitochondrial membrane is an integral protein and antiproter. which transports acyl carnitine inside the inner mitochondrial matrix and transports out same mole of carnitine. Then acyl carnitine reacts with coenzyme A, another enzyme, acyl carnitine transferase 2, present on the inside of the inner membrane of mitochondria, catalyzes the reaction to reform acyl coenzyme A and carnitine within the inner mitochondrial matrix. Now fatty acid is available within the mitochondria for oxidation. Now how fatty acids are oxidized within the mitochondria? To understand the biological degradation of fatty acids, German biochemist F. Nook carried out an experiment. He used phenyl residue as a reporter group because it is not metabolized and excreted in urine. Phenyl substituted odd and even chain fatty acids were given in diet of the experimental animal, dog or rabbit. When rabbits or dog were fed an even carbon fatty acids of different chain length labeled with a phenyl group at the omega carbon, phenyl lactic acid was excreted in the urine. When rabbits were fed odd chain fatty acids with omega phenyl derivatives, benzoic acid was excreted. From this result, it was understood that fatty acids are oxidized stepwise at its beta carbon and releasing 2 carbon units acetic acid at each step. Fatty acyl-CoA within the inner mitochondrial matrix undergoes only 4 steps to produce 2 carbon unit acetyl-CoA: oxidation, hydration, again oxidation, and thiolation. Each cycle of beta oxidation produces 2 carbon unit acetyl-CoA, and the carbon chain length of the fatty acyl-CoA reduces by 2 units. Lastly, butynyl-CoA, a 4-carbon acyl-CoA, is produced by beta oxidation of even chain fatty acid and undergoes last cycle of beta oxidation. If it is a odd chain fatty acid, final product is propionyl-CoA and does not undergo beta oxidation, and it is converted to succinyl-CoA, a TCA cycle intermediate. Acyl-CoA within the inner mitochondrial matrix is oxidized by the enzyme adjacent to the respiratory chain in the inner mitochondrial membrane, collectively known as fatty acid oxidase. There are different dehydrogenases, hydrolases, and thiolases. In the first step, acyl-CoA dehydrogenase catalyzes enzymatic dehydrogenation at the alpha and beta carbon to form delta-2 trans-enoyl-CoA. Coenzyme for the dehydrogenase is a flavoprotein containing FAD as a prosthetic group. Hydrogens from alpha and beta carbons are transferred to FAD to yield the reduced form of FADH2, which denotes its electron to the respiratory chain via a second flavoprotein, electron transferring flavoprotein, which in turn passes the electron to the respiratory chain. Acyl-CoA dehydrogenase also possesses a chain length specificity. There are different long chain, medium chain, and short chain acyl-CoA dehydrogenase. A genetic deficiency of medium chain acyl-CoA dehydrogenase decreases the beta oxidation of medium chain fatty acids. Then medium chain fatty acids are oxidized by omega oxidation to form dicarboxylic acids, which are eliminated in large amounts in the urine, is known as dicarboxylic aciduria. Step 2. In the second step, delta 2 trans-enoyl CoA is hydrated to form 3-hydroxyacyl CoA by the enzyme enoyl CoA hydratase. The addition of water across the delta 2 trans double bond is stereospecific and results in the formation of ill stereoisomer of 3-hydroxyacyl CoA, which is absolutely specific for second dehydrogenation step. This reaction has no chain length specificity, no cis-trans specificity, but D isomers produced from the delta 2 cis or delta 3 trans unsaturated acyl CoA could not undergo second dehydrogenation step. Step 3 or second dehydrogenation step. L3-hydroxyacyl CoA is dehydrogenated to form 3-ketoacyl CoA by 3-hydroxyacyl CoA dehydrogenase, which is specific for A stereoisomer but has no chain length specificity. In this reaction, NAD+ is the coenzyme and acts as a specific electron acceptor. NADH is formed in this reaction, which donates its electron equivalents to NADH dehydrogenase of the mitochondrial electron transport chain to produce energy. Last step of beta oxidation is step 4, the thiolysis or the cleavage step. In this step, acetyl-CoA acetyltransferase, or more commonly known as thiolase or beta-ketothiolase, catalyzes the cleavage of 3-ketoacyl-CoA by the interaction with free coenzyme A to yield the carboxyl terminal 2-carbon unit as acetyl-CoA. The remaining fatty acids shorter by 2 carbon atoms, acyl-CoA is formed and enters the next beta oxidation cycle.